What if the cutting edge cure for brain cancer is actually, teaching the tumor how to build a better fortress?
Yeah, it's a terrifying thought.
It really is. Today, we are looking at an unprecedented biological war zone. We have a landmark August 2026 paper published in the journal Cell by an incredible team of researchers out of the University of Pennsylvania and Institute Curie.
Right, an amazing team.
Led by Nelson F. Freeberg, Zeve Binder, Dana Silverbush, and others. This team managed to pull off something just extraordinary. They gave us a real-time microscopic look at exactly how the human immune system and recurrent glioblastoma tumors evolve when we inject engineered SRRT cells directly into the fluid of the human brain. Welcome to the deep dive. We are executing a massive mission today for you, looking at how to map a constantly shifting battlefield.
It's a phenomenal paper. I mean, what we are looking at here is a masterclass in reading a dynamic, evolving ecosystem. A tumor is not a static target.
Right. It's not just sitting there.
Exactly. Treating it isn't just dropping a bomb and walking away. It's a siege. And both sides are adapting in real time.
Okay, let's unpack this for a second. Instead of a standard IV drip where you're just hoping the medicine crosses the blood-brain barrier, they are using intracerebroventricular SAR T-cell therapy.
That was a mouthful. But...
Yeah, right. But essentially, they're injecting these engineered killer cells, which are basically the patient's own immune cells reprogrammed to hunt specific cancer markers, directly into the cerebrospinal fluid, straight into the ventricles, deep inside the brain.
Straight into the fortress.
Exactly. We are dropping the special forces right into the courtyard of the castle. But they didn't just dump the cells in randomly. They designed a highly structured trial with two very different dosing strategies.
Right, the cohorts.
Yes, the cohorts. We have a high-dose discovery cohort, patients P1 through P8, and they specifically focus their data on P1, P4, and P7. Those patients received a massive dose of up to 25 million CRT cells.
A huge amount.
And then a high-dose validation cohort, P9 through P12, getting that same dose. But then they had a low-dose cohort, patients P13 through P18, with deep data on P13, 14, and 16, who received exactly 5 million cells. They tracked these patients at day 0, day 7, day 21, and then followed up with fluid withdrawals for months.
Mm-hmm.
But wait, I have to ask, if you're injecting up to 25 million hyper-aggressive engineered cells into a closed system like the human skull, wouldn't the resulting inflammation just be completely fatal? I mean, why are they risking doses that high?
Well, the rigid casing of the skull is exactly why they had to structure the trial this way. Think about it. If you get a cut on your arm, your immune system rushes in and the arm swells up.
Right, it gets red and puffy.
Yeah, and it's annoying, but it's fine. Your skin stretches. But if you introduce millions of engineered ART cells directly into the brain fluid, they're going to find their target and trigger massive inflammation. And inside a rigid skull...
swelling has nowhere to go.
Exactly. It compresses brain tissue, which is a life-threatening emergency. So the researchers had to test tolerability alongside immune scaling. They needed to find out, does 25 million cells provide 5 times the therapeutic cancer-killing effect of 5 million cells? Or does it just cause 5 times the dangerous intracranial pressure?
So the balancing act.
It is. They were meticulously mapping that threshold between maximum efficacy and catastrophic swelling.
Okay, let me make sure I have this locked in before we move forward. Let's do a quick bullet summary for you listening. Bullet 1. To bypass the fortress walls of the blood-brain barrier, researchers injected engineered CAR-RT cells directly into the brain fluid. Bullet 2. They rigorously tested a low dose of 5 million cells against a massive high dose of 25 million to find the exact line between destroying the tumor and causing fatal brain swelling.
Perfect summary.
Thanks. Now, here is a question for you in the audience to think about. Think about your own body's natural defenses. If you suddenly introduce millions of foreign, hyper-aggressive engineered cells into your brain fluid, how do you think your natural, native white blood cells are going to react? Will they get out of the way, or are they going to join the fight?
And that question brings us to the single-cell RNA sequencing data, which honestly is where this paper completely changes the game.
The cRNA-seq. This is how they actually tracked what the native immune system was doing, right?
Yes. And, to understand the gravity of what they found, you have to understand the technology. Traditional biopsies are, they're like listening to the roar of a stadium crowd from outside the arena.
Okay.
You know a game is happening, and you hear general noise, but that's it. Single-cell RNA sequencing is like putting a microscopic microphone on every individual fan inside that stadium.
Oh, wow.
Yeah. You know exactly who is cheering, who is booing, who is buying a hot dog. The machine isolates individual cells, cracks them open, and reads the specific RNA transcripts, basically the genetic instruction manuals, that the cell is actively using in that exact moment.
That is staggering. So when they put microphones on all these cells in the brain fluid, who exactly was in the stadium?
An entire microscopic city. I mean, they tracked exactly 28,127 malignant tumor cells. But they also mapped the entire immune neighborhoods surrounding the tumor.
Like what?
They found CD4 and CD8 T cells, regulatory T cells, macrophages, monocytes, neutrophils, B cells, plasma cells, microglia, glial cells, and neurons.
Wow.
Yeah, every single one of those distinct cell types was captured and analyzed.
Let's focus on the CD8 T cells for a second, because these are the body's native killer cells, right? Like the local city militia that was already there trying to fight the tumor before our CAR T cell special forces arrived. How did they react?
Well, what's fascinating here is that the researchers zoomed in on what they call TCR-matched CD8 T cells. These are the specific native immune cells that already have the genetic receptors to recognize the tumor.
So they already know what the bad guy looks like.
Exactly. And when they compared the state of these cells before the TRT infusion to their state after the infusion, the data showed a massive biological shift. The native cells essentially leveled up. Their cytotoxicity score, which is the mathematical measurement of how lethal they are, it just shot up significantly.
Wait, I need to understand the how here. How does a cell objectively become more lethal? I mean, what is the mechanism behind that score?
It comes down to the specific weapons the cell is manufacturing. The sequencing showed a massive upregulation in a gene called GZMH.
GZMH?
Right. And this gene gives the instructions to produce granzyme H. You can think of granzymes as molecular grenades.
Grenades.
Yeah. When a T cell attacks a cancer cell, it first uses a protein to punch a microscopic pore, like a little hole, in the cancer cell's membrane. Then it drops granzyme H through that hole. Once inside, the granzyme triggers a self-destruct sequence in the tumor cell.
That's brutal and awesome.
It is. And the CRNA-seq also showed a spike in IFNG, which produces interferon gamma. Now, this isn't a weapon. It's a chemical radio signal.
Okay.
It screams out to the rest of the immune system, forcing other cells to become more aggressive and altering the battlefield to make it harder for the tumor to hide.
I love a good visual memory trick. Imagine a radar screen sweeping the battlefield. The cellular T cells drop in, and it's a massive blip on the radar. But as the radar sweeps again, it shows the native CD8 T cells, the local police force, literally glowing brighter. They are manufacturing more granzyme grenades and screaming on the interferon radio.
That's a perfect way to visualize it.
But here is my concern, right? If you sprint at full speed, you eventually collapse. Don't these native cells burn out from fighting so hard?
And that is the most remarkable part of the finding. Normally, yes. It's a phenomenon called T-cell exhaustion. When native immune cells are exposed to a stubborn tumor for too long, they get overstimulated. They start expressing inhibitory receptors, basically biological brake pedals that shut them down.
Right.
But the sequencing proved that their exhaustion score stayed completely stable. The researchers specifically looked for weakness in exhaustion markers, like the genes REG4 and LRN3. And they were essentially absent, or just totally flat.
Wait, really? So the special forces didn't just fight. They acted as a catalyst that permanently trained and buffed the local militia, making them more lethal without draining their batteries.
Exactly.
Let's pull this together with another mini recap. Billet 1. Using single-cell RNA sequencing, researchers essentially put a microphone on tens of thousands of individual cells to read their real-time genetic activity. Bullet 2. After the CRT infusion, the body's local immune cells evolved. They upregulated genes for molecular weapons like granzyme H and communication signals like interferon gamma, becoming highly cytotoxic without increasing their exhaustion markers.
Spot on.
Now, here is a question for you listening to ponder. If the engineered CAR T cells are attacking and the local immune cells have leveled up their weapons and are fighting harder than ever without burning out, why isn't the tumor completely wiped out? What on earth is glioblastoma doing to survive a coordinated attack like that?
And that question brings us to the terrifying genius of this specific cancer. I mean, if the immune system is a dynamic, evolving force, the tumor is an equally dynamic, adapting fortress.
It's fighting back.
Oh, absolutely. The researchers analyzed those 28,000 malignant cells and discovered the tumor executes a highly coordinated three-pillar defense mechanism called a mesenchymal shift.
Okay, let's break that down. What actually is a mesenchymal shift? It sounds like the tumor is shape-shifting or something?
That is exactly what it's doing. It's a process known as epithelial mesenchymal transition, or EMT. In cellular biology, epithelial cells are like bricks. They stay in their assigned places, they form neat protective layers, and they are rigidly bound to one another.
Okay, bricks. Got it.
But mesenchymal cells, on the other hand, are more like aggressive amoebas. They lose those rigid anchor points, they alter their internal cytoskeleton, and they become highly mobile, fluid, and adaptable. So when a tumor undergoes EMT, it drops its rigid brick structure and turns into a heavily armored, shape-shifting monster.
That is terrifying. And the paper outlines 3 specific pillars of this transition. So what is the first pillar the tumor relies on?
Pillar 1 is a literal physical barrier. When the CAR T cells attack, the single-cell data shows the tumor immediately upregulates genes like COL6A2, CD44, and PUSTN.
How do those genes stop a killer T cell, though?
Well, they hijack the body's cellular scaffolding system. COL6A2, for example, instructs the tumor to start rapidly synthesizing and pumping out thick, fibrous collagen into the extracellular matrix, which is the space between the cells.
Wait.
Yeah, they're essentially pouring biological concrete. The T cells get physically trapped in this dense, sticky web before they can even get close enough to use their granzyme weapons.
A microscopic concrete wall. That is wild. Okay, what is the second pillar?
Pillar 2 is metabolic armor. They upregulate a specific gene called SLC16A1.
Let me guess, this changes how the tumor eats or breathes.
Close. It changes how it exhausts waste. When immune cells and tumor cells fight, they burn through massive amounts of energy using a process called glycolysis. And a byproduct of this is lactic acid.
Okay, acid.
Right. The battlefield becomes a highly toxic acidic swamp. Normally, this acid would poison and kill the cells. But the SLC16A1 gene creates a specialized protein channel, a high-powered exhaust vent. It allows the tumor cells to constantly pump the toxic acid out of their own bodies and into the surrounding environment.
So they survive the acidic siege while the T cells trying to attack them suffocate in the toxic microenvironment.
Exactly. They weaponize their own waste.
So they are building concrete walls and wearing metabolic hazmat suits. What is the third pillar?
The active immunosuppression. This is where the mesenchymal shift truly takes over. They utilize factors like VEGF-A and a master regulator gene called TWIST1. TWIST1 acts like a commanding general. It's a transcription factor, meaning its whole job is to go into the DNA and turn entire networks of other genes on or off.
Wow.
TWIST1 orders the tumor cells to fundamentally change their identity and start emitting chemical signals that actively put the immune system to sleep. They aren't just hiding behind a wall. They are throwing tear gas over the wall to incapacitate the attacking T cells.
Okay, wait, let me pause you there. I need to push back on this.
Sure.
We are looking at a progression of events, right? We inject the CAR T cells, and later we withdraw fluid and see that the tumor has built walls, put on hazmat suits, and started emitting sleep gas. But glioblastoma is an aggressively mutating cancer by its very nature. How do we know the CAR T cells actually caused this shift? Couldn't the tumor just be naturally getting worse, and it has absolutely nothing to do with the therapy we gave them?
That is the exact critical distinction the researchers needed to make. Observational data in a human patient shows correlation, but it does not definitively prove causation.
Right.
To prove that the CAR T cells directly caused the shape-shifting, the researchers had to take this out of the human body and into a strictly controlled laboratory environment. So they built GBM organoids.
Organoids? Are these just like tumor cells smeared on a flat petri dish?
Not at all. Flat petri dishes don't accurately represent human biology. Organoids are three-dimensional living microtumors. They take actual patient cells and grow them in a gel matrix, so they form a complex spherical architecture, just like a real tumor inside a brain.
Okay, so it's a 3D model.
Yes. They then set up a highly controlled experiment. They co-cultured one set of these living organoids with the specific EGFR 806 CAR T cells used in the clinical trial. And then they cultured a second set with a control group of CD19 CAR T cells, which are designed for leukemia and completely ignore glioblastoma markers.
So one tumor gets actively attacked and the other tumor is just sitting next to harmless cells.
Precisely. And the data was undeniable. The control group tumor did nothing. But the tumor attacked by the EGFR targeting CAR T cells immediately triggered the hallmark epithelial mesenchymal transition.
Wow.
The organoids prove that the tumor doesn't just randomly put on armor. It actively recognizes the specific engineered weapon being used against it, and it dynamically alters its own biology to survive that exact attack.
That is an incredible piece of scientific detective work. Okay, let's do a quick synthesis to lock down the tumor's counterattack. Bullet 1. Glioblastoma actively defends itself through a mesenchymal shift, abandoning its rigid brick structure to become a fluid, adaptable, amoeba-like entity. Bullet 2. We know exactly how it defends itself. It pours biological concrete using collagen genes like CL6A2. It equips metabolic exhaust vents using SLC16A1 to survive toxic acid. And it uses master genes like TWIST1 to put the immune system to sleep. Bullet 3. By using 3D living organoids, scientists definitively proved the shape-shifting is a direct causal reaction to our CAR T-cell therapies.
Beautifully summarized.
Now here is your final question to think through for you listening at home. If you are an oncologist and you now know with absolute certainty that the tumor is going to automatically build a wall and put on metabolic armor the second you send in your special forces, how would you change your strategy for the next wave of the attack?
That question represents the absolute bleeding edge of modern oncology, because we have mapped the enemy's playbook.
We really have. When we synthesize everything we've looked at today, the picture of glioblastoma treatment completely changes. We are not dealing with a simple biological equation. Delivering LRT cells directly into the brain fluid is a brilliant delivery mechanism that acts as a massive catalyst. It effectively scales up the endogenous local immune system, upgrading native T cells to become incredibly lethal without burning them out.
Exactly.
But simultaneously, the tumor recognizes the specific threat and dynamically shifts its physical shape, alters its acid metabolism, and erects physical barriers to induce immunosuppression and survive.
And, it's crucial to recognize that as we look at this data, we're simply reading the biological reality of clinical trial participants and meticulously controlled laboratory models. We aren't endorsing a specific brand or anything. This paper is a completely objective readout of cellular biology. It maps the harsh, complex reality of tumor evolution without bias. We are observing the mechanics of survival at a microscopic level.
We are just reading the map they've drawn for us. So where does this new map lead us?
Well, it leads to a fascinating and deeply provocative thought, which directly answers the strategic question you just posed. If it is a biological fact that these tumors automatically trigger this mesenchymal shift, if we know they're going to deploy TWIST1 to suppress the immune system and SLC16A1 to vent toxic acid, then maybe the future of cancer therapy isn't about trying to engineer an even stronger, more aggressive CAR T-cell.
Okay, what is it about then?
Maybe the future requires a Trojan horse approach.
A Trojan horse, like a multi-stage siege?
Exactly. Imagine administering a highly targeted pharmacological drug first, a drug whose only job is to slip into the tumor and chemically block the TWIST1 gene or permanently weld shut that metabolic exhaust vent.
Oh, wow.
Right. You administer the Trojan horse to completely strip away the tumor's ability to change its armor, and then you inject the CAR T cells into the brain fluid. We disable the fortress's automated defenses before the special forces ever arrive at the gate.
Disable the defenses, then drop the payload. That is a brilliant and completely logical evolution based on the mechanisms we've explored today. It really makes you appreciate not just the terrifying complexity of the human body, but the relentless ingenuity of the scientists working to crack these biological codes.
It's an ongoing war, but we're getting smarter.
We absolutely are. We started this deep dive by talking about how disease used to feel like a simple binary. You take a pill, you get better. But now we see that inside the fortress of the human brain, the walls are constantly moving, the local militia is always adapting, and the war is being fought gene by gene, cell by cell. Thank you for joining us on this deep dive. Keep questioning the world around you, keep looking for the how and the why behind the headlines, and we'll catch you next time.